So without further ado, I'd like to introduce Dr. Lisa Schimmenti, who is a clinical geneticist at Mayo Clinic. Dr. Schimmenti's research is focused on genetic conditions associated with hearing and vision loss, conducting clinical research and studies involving zebrafish to model human conditions. And Dr. Schimmenti, over to you.
Well, I want to start by thanking the organizers for inviting me because it's been a real honor to be here today. And I'm just blown away at all the good that has been going on this morning and sharing all the good things that everybody is developing. So I want to start with talking about the genetics of Myhre syndrome and the objectives of this talk. Individuals should be able to describe how DNA is transcribed to RNA to make a protein, to understand that variants in the DNA code lead to changes in protein formation, to be able to relate how errors in the DNA code cause abnormal activation of the protein SMAD4, and then to understand the importance of research to answer unanswered questions and provide potential treatments for Myhre syndrome. And you heard a lot about that this morning from Dr. Lindsay. So we're going to start by getting really small. And I want you to think about yourself as a collection of cells. And so in this drawing, this is a cardiomyocyte. This is a hair cell in the inner ear. This is a neuron. This is a defender cell. And in every cell, there's a nucleus. And in the nucleus is DNA. The DNA that we have forms our genome. And DNA in our cells is a double-stranded molecule that I'll show you some pictures of. It's made of chemical bases. And these chemical bases are A, T, G, and C. There are 6 billion chemical bases in each of our cell. We get 3 billion from each parent. And the collection of all these chemical bases is called our genome. This is a picture from the NIH. This is showing the double-stranded DNA molecule. The DNA molecule is made of a sugar phosphate backbone that holds all the chemical bases in place. And A binds with T and C binds with G in the DNA double-stranded helix. And so we'll talk about A, T, C, and G in detail. So our DNA is organized into genes. There are 20,000 genes in the genome. The primary role of each gene is to provide instructions for a cell to make protein. There are other jobs that genes do, but the main role and the role that's germane to Myhre syndrome is making a protein. So each gene is made of a code of A's, T's, C's, and G's. And when we do genetic testing, when we sequence a gene, we sequence all the A's, T's, C's, and G's in a specific order. So DNA is essentially the boss. DNA holds the code. And it's a triplet code. And it's essentially like an alphabet that only has 4 letters. All the words in this alphabet are only 3 letters long. And so this triplet code provides instructions for the cell to make RNA. And RNA is the messenger. So if you think about DNA as the boss now talks to RNA and says, hey, RNA, your job is to now take my information that I'm giving you and then go somewhere in the cell and make a protein. And so this is an example of a DNA code and an RNA code. You'll notice all the T's are changed to U's. And the reason for that is you don't want the RNA code hanging around forever. And so the U's make the RNA unstable so that it is just useful for making protein, and then it gets destroyed by the cell. The cell does a recycling job and cleans it up. But the DNA is really sturdy and it will hang around, and it then is available for being passed down to the next generation. So DNA to RNA. So the next thing that happens is that the RNA then goes to a special part of the cell. It's called a ribosome. And that ribosome is a factory. And that factory then takes the RNA code and places an amino acid, which is a building block of protein. in the correct position for the code. So in this example, the first three letters are ATG, that gets transcribed to AUG and then translated to a protein. And every protein starts with methionine. And then we just go down the line until we make a whole protein. So this is like a little tiny protein that we made. And then here there's a stop. So the DNA code can't go on forever, and the protein is a specific length. And so there's actually a code to you could stop now. You finished doing your job. You made your protein. This is called the central dogma of molecular biology. And so here's DNA starting with an ATG. Here's a longer stretch of DNA. Here's the triplet code. Here's the RNA. And then here's the protein that's produced from that RNA in the cell. I want you to remember the central dogma because it's really important. And here's the central dogma. This is Luna. Some of you know Luna. She's Patty Schultz's dog. Thank you, Luna. All right, so what can go wrong? I'm just using regular words as an example, and I'm just using 3-letter words. We do have more than 4 letters here. And so this would be a typical, say this is a typical DNA sequence. The big red dog ran out. If in this word, the E was changed to an A, that would be a missense. And so instead the meaning is changed. And this is the big rad dog ran out. If we delete one letter from this sentence, like in this case, the I in big, we end up with the triplet code shifting over one. And so now this sentence is the big ad ogre ano, and that doesn't make any sense to the cell. And so it will make this goofy protein that may not have any function at all. And generally what cells do is they will just get rid of that protein or get rid of the RNA that codes for that protein because it just doesn't want stuff around it doesn't need. And then the same thing's true if there's an insertion. So in this case, there's 2 O's in dog. So we end up shifting the phrase and it ends up being the big red du gras-nu. Again, this is not a very useful protein for the cell. So when we do genetic testing, we isolate DNA from blood, saliva, or other tissues. We then take that DNA and put it in a sequencer where all the A's, T's, C's, and G's are sequenced. They're put in the correct order. And then we take the computer readout from the genetic testing and we compare that to typical. And typical is basically a database of sequence information from a number of individuals who've been sequenced who supposedly have no significant health issues. Any deviation from typical gets flagged by the software, and then flag sequences are evaluated. And so when you get your report back, you get all the flag sequences back. And so when we get our genetic testing reports back, we can find different things. And let me just talk about some different types of genetic testing. The first is single gene sequencing. That means that the physician that was evaluating you or your child said, you know, I think this person has Myhre syndrome. I am just going to sequence one gene, and that's SMED4. We could also do panel sequencing where there's a panel of genes that are associated with heart disease, and there are usually a couple of hundred genes here. Then there's exome sequencing, and this is where we sequence all 20,000 genes in the genome, but leave behind all the other DNA sequence to sequence. And now we're really moving into a new space where we're doing genome sequencing. So we're sequencing all 3 billion base pairs in a cell. And it's turned out to be really exciting because we're actually finding things we didn't find before, or finding in some people other conditions that we didn't expect. And so when the results come back on this testing, there are 5 possible results. We'll start in the green. You sometimes can get benign or likely benign. sequence variants, and those are variants that the lab sometimes won't even report out. Every once in a while they do, because they may feel there's something important to discuss. There's also likely pathogenic and pathogenic variants. Those are what we used to call mutations, and those are DNA variants that are well established to be causative of a condition. And then it's not infrequent that suddenly you see a list of variants of uncertain significance in the report. And these variants are gene sequences that are different than what we expect to see in that DNA, but there's not enough evidence to support that that variant is causing a condition or not. And some of you may have experienced this when you received your genetic testing results. So let's focus on SMAD4. This is one of 20,000 genes in the genome, and the single variant in SMAD4 is 1 in 3 billion. And so the odds of this happening is really small, and explains why Myhre syndrome is so rare. And so one of the unusual things about Myhre syndrome is that there's a specific 3-letter code in Myhre syndrome that seems to be changed most often, and that's ATA, and it can be changed in different ways. So I'm going to start with the most common. At position 1498, there should be an A present, and instead there's a G. And so it changes the triplet code from the amino acid isoleucine to the amino acid valine. And that's the most common variant we see in individuals with Myhre syndrome. We can also see at position 1498, there should be an A, but instead there's a C. And so that changes an isoleucine to a leucine. That's the rarest. We could also see, instead of ATA, we could see CCA, and that now changes an isoleucine to a threonine, again, changing the code, changing the protein. And these amino acids are different from each other chemically. And so this changes the way SMAD4 works. And so here's ATA and ATG. So at position at 1500, there normally should be an A, and instead there's a G. And that changes an isoleucine to a methionine. And then a few other nucleotides away, so a few other A's, T's, C's, and G's away, is this variant at 1486. Normally there should be a C there, and instead there's a T. And so it changes an arginine to a cysteine. And all of these change the way the SMAD4 protein works. So the variants in SMAD4 in Myhre syndrome are activating variants. And this is different than other conditions, and we'll talk about that. But basically, these variants change the way SMAD4 works. The other thing that's really important, because when you got your report and you Googled SMAD4, you may have seen these other conditions, like juvenile polyposis, or hereditary hemorrhagic telangiectasia, or hereditary hemorrhagic telangiectasia plus juvenile polyposis, or even what they call somatic mutations within specific cancers. Your people with Myhre syndrome who have SMAD4 variants are not at risk for these other conditions, and they only have one variant in SMAD4. So you don't have your SMAD4 is not dynamic. It's not changing. You have the SMAD4 variant that you started out with and have it for your whole life in the cells of your body. So this is also what we call an autosomal dominant condition. So we have 2 of every gene, one from our mother and one from our father. And in a dominant condition, only one copy of the gene has a variant. The other copy of the gene has a typical sequence. And so SMAD4 variants are dominant, meaning you only need one copy of the gene to have the condition. And then, by extension, individuals who have a SMAD4 variant and have had children have a 50% chance of passing this variant to their children. But for many people with a SMAD4 variant, this is a de novo change, and it means a new genetic change. And it's because every time a cell in our body divides, DNA needs to be copied. And the copying process for DNA is imperfect. It's like if you were given a paragraph to copy and you suddenly spelled a word wrong in that paragraph. Our cells do the same thing. And if that DNA change, that missense change occurs in a specific location in SMAD4, then a child will have Myhre syndrome. I want to make it clear that there's nothing that anyone has done that caused this to happen in their children, and nothing that happened before or during pregnancy could prevent this. And that's really important because I think for all of us who have children, if there's something going on with our child, we blame ourselves. And I want to make it clear that this is not something that you need to blame yourself about. We don't have control over the way our DNA copies itself. So you've probably seen versions of this figure before from lots of people. This is kind of a simplified version. And so the job of SMAD4 is to transmit signals from the cell surface receptors to the nucleus. And so this is kind of a schematic of a cell. This double dotted line is actually the cell membrane. And the 4 structures you see here that look like they're cradling a molecule are receptors. It's almost like these receptors are going to a baseball game and they're holding out their glove and they're waiting for a ball to enter their glove. And when the ball enters the glove, the receptor then phosphorylates SMAD2/3 in the TGF beta side. On the BMP side, it's SMAD1, 5, and 8. SMAD4 then hops on with the other SMADs, and then they get transported to the nucleus of the cell, where their job is to then tell other genes to turn on and make RNA and make other proteins. And this is kind of cool, and it goes back to what Dr. Lindsay was saying, in that these proteins are functioning differently. So with somebody with hereditary hemorrhagic telangiectasia or juvenile polyposis, there's actually a deletion within the SMAD4 gene that leads to reduction of the SMAD4 protein. And so it's almost like this SMAD4 is whispering, there's not enough of it to do its job. However, in the typical situation, the SMAD4 is talking at a normal volume. But in Myhre syndrome, SMAD4 is overactive. It's just doing much more than you want it to do. So it's basically overexpressing other genes in the genome. And so part of the strategy that Dr. Lindsay and others are thinking about is, how do you just tell SMAD4 to just kind of tone it down a bit? And you don't have to be overactive. And so this leads us to the research questions. And for all of us, we ask, how does activation of SMAD4 lead to the various physical findings in Myhre syndrome? And could we slow down the overactive SMAD4 and change the outcome for patients with Myhre syndrome? And then how does cell and animal models provide a resource to study methods to modify overactivity? And you've seen that this morning. And so I think this is my last slide. So I hope I fulfilled the objectives of this talk today. I hope that this was pretty clear. If we have time and Kate and Inez feel like it's okay, I'm happy to answer questions. And thank you for your time today.
Thank you, Dr. Schimmenti. We do have a few questions, so hopefully you don't mind answering them. Can we still hear you?
Yes.
Okay, wonderful. Our first question came fairly early on in your presentation. The question was, what is the PGD-FRA gene?
So, Pete, let me bring back was it the slide early on?
Yes, it was one of your first slides, I believe.
Huh, okay. It
was early on.
Lisa, it's platelet-derived growth factor receptor alpha.
Thank you.
It's a receptor tyrosine kinase.
Thank you so much.
It causes gastrointestinal stroma tumors. It doesn't have a clinical syndrome phenotype.
Okay.
Okay. I think that might answer the question. Are you still looking at your slides, Dr. Schimmenti?
Yeah, I'm trying to see where it is, but that's okay. Just keep going.
Yes, we'll keep going. And we can always come back to it.
I don't think it was on the slide. I don't know if the questioner might want to explain why. It does not cause Myhre syndrome. So I'm
Okay.
not sure where the question is coming from, but it might help if they're willing to disclose.
Okay. All right. We'll have the person who asked the question elaborate a little bit more in the chat for us. So we can move on to the next question and we'll come back to that one. How common is it to have multiple variants of unknown significance or other conditions that might show up on a genetic test?
Yeah, it's really common. For those of you who are listening today who've had their genome sequenced or had an exome sequenced, your report might have said, oh, there's a SMAD4 variant, it's a pathogenic variant. And then they give you a list in a separate table of all the variants of uncertain significance. So when we do this sequencing, yes, we could sometimes find somebody has more than one condition, and it's something we've seen before. And we're learning more about this. And can it influence, one of the questions that we always ask is, can these variants influence Myhre syndrome? Like, why are some people with Myhre syndrome very, very verbal and very gifted and really talented, and other individuals may have autism and have decreased speech, and we don't know at this point. So I think there's a lot to learn. So thank you. I have a diagnosis of PDGFRA gene variant. Okay, and it might be, since that gene is not associated with any disease. I was reading the question in the chat. I don't think we know what it means, especially if the report says a variant of uncertain significance rather than a pathogenic variant.
All right, thank you. Thank you so much. All right, so my next question is, do we know anything about the variations in the SMAD gene and how it expresses itself in different patients?
So most of my experiences with adults who have two specific variants, I'm going to ask Angela to hop on and talk about the patients with the most common variant.
Okay.
So
what is the specific question?
Oh, the question is the difference in phenotypes. And I know you've done work on this, Dr. Lin, on patients who have the most common variant and why there's such differences in phenotypes. So that's going to punt the question to you.
We've looked very closely at our 47 patients. When I say we, I need to thank Eleanor Simone for this. The one thing that we can say is those individuals, some of you who are listening, who have the arginine 496 to cysteine variant are much more likely to have normal height, completely normal. And whereas the other variants are more likely to have growth retardation, small gestational age, small size, growth restriction. These are not completely separate, but this is what I'm saying is more. There's some other features that I think I can share, which is in terms of hearing loss, it seems to be more common in those who are the isoleucine 500 valine variant. There's some other features that we're delineating in terms of the heart, but we're not quite ready to share them yet. The problem is we need as many patients as possible. And when we start to do comparisons, I do what I think somebody else did, I don't know if it was Mark. I generally split just into two, the arginine 496 to cysteine variant versus the isoleucine 500 valine. That doesn't mean that the other variants are not important. I think Lisa herself, though, could speak to the variant change from isoleucine to leucine, which she did include on her slide. I'll let you
Right.
speak to that.
Yeah, and that's our rarest variant. And I probably have the most experience with that right now. That individual is brilliant and talented and has all the other features of Myhre syndrome. But I don't, how that variant is different. Dr. Lin did some analysis, some bioinformatic analysis with a colleague, and found that variant may have a milder impact on the function of SMAD4, but that's just a computational analysis, and it was something that we need to test that hypothesis in cells or model organisms. But at least that information is, it at least computationally, it suggests that variant may be different.
Oh, it helps if I unmute myself. I'm talking, and you can't hear me. So one last question on this particular topic is, do you think, and this is kind of a broad question and you may not be able to answer it, and that's okay, is do you think we'll see Myhre diagnosed in positions other than 496 and 500?
That's a really good question. I think if we did, there's a technique one can do where you could model variants in a gene and basically computationally change every variant and see if it has a potential effect on a protein. So I think, yes, it's possible we might see other variants, but I think now that we're doing sequencing of every gene and sequencing the entire gene, we may stumble upon these. And then we have to see if that person has Myhre syndrome or not. Do you want to hop in, Angela? Because I know this is a discussion we both have had.
Yeah, no, these are all good questions. I would say just in general to the community listening, if there's anybody who hasn't had testing yet or hasn't had recent testing, we can help people find access really around the world. Geneticists are a small, friendly community, and we would help.
Very true, very true. I have a technical question now.
Okay.
So I'm hoping I'm saying this correctly. It says, does epigenetics and lifestyle habits influence phenotypic characteristics in Myhre syndrome patients?
Yeah. So epigenetics refers to the alteration of the way a gene is expressed in the genome. So, let's go back to the central dogma. DNA then makes RNA that makes protein. And if there are modifications to the DNA that basically slow down the production of RNA, you can have a different outcome. We're just kind of getting into that. Yes, there's tons of things on the internet that if you drink certain things or eat certain foods, it's going to change the epigenetic profile of your DNA. That's still kind of hand waving. There's not a lot of hard science behind that. Yeah, I think it's a really good question, and it's things we're looking at for genetic conditions where we don't find any DNA sequence changes, and we're actually doing methylation sequencing, which is one of the processes for silencing genes or slowing down genes. But I don't think this is ready for prime time yet, or I could give you any advice that's helpful.
But we would congratulate the questioner that it is a very valid area of research, epigenetics. Yeah. In fact, that's valid for almost everything. We look for ways to explain differences. We can have something coarse like the particular variant, but then why within the variant are people different? terms of lifestyle habits, there is not a known lifestyle or out, and we speak of risk factors, folks, by the way, very different from causation. So we don't know a risk factors such as smoking, alcohol, diabetes. We are looking though at something that might be considered a risk factor, which is age. So we look at maternal age and paternal age. We generally see maternal age as a risk factor for the conditions that are associated with an extra chromosome, aneuploidy, trisomies, and then for advanced paternal age, we already know that's a risk factor for some genes that are associated with this type of inheritance called Mendelian. And that's research coming, so we don't have an answer today yet.
Right. And they also want to make it clear that I don't want anyone to feel guilty for choices that they made because we don't know enough. Yeah, I've had parents beat themselves up because they took a wonderful vacation and had a bunch of drinks and got pregnant. And then their child had a condition that didn't cause this. So please, don't go home and think you did something.
And I see the questioner asking about publications on the variants and descriptions of their outcomes, and that's coming.
Publication is coming for.
Yes, this is our MGH series, and it's quite long. It's taken years to work on, but we're still hoping to get it completed.
Thank you for answering that question. I'm so glad that we have time for a couple of more questions. One of the questions that came across is, how do we tell new doctors and friends and relatives what has happened in genetics with Myhre?
Yeah.
Like a short sentence that you can explain to friends and doctors on about Myhre.
Yeah, no. So let's start with doctors. Generally, when you start with a new physician who's going to take care of your child or your family, and you tell them that your child has Myhre syndrome, and you share something, some things that are really pretty straightforward to share is the Medline Plus from the NIH. It's pretty short. There's also the gene reviews. There's also the information from the Myhre syndrome website. And if that physician gets excited and says, I'm really looking forward to taking care of your child, you're in a good place. If that person starts to glaze over and back up, maybe you might want to think about another place. The other thing about genetics is that one of the reasons for following up with genetics is that our services, our role is to help with management. And so the primary care provider doesn't have to do everything. But for certain things, we generally make lists of all the Myhre management things that need to get done and follow up and collaborate with physicians. I mean, I'm pretty lucky, and so is Dr. Lynn. We work in places where we have colleagues who are now the world's expert in Myhre syndrome and diabetes, or Myhre syndrome and pulmonology. And so we all communicate with one another and make sure that this multi-system condition is being managed. And so I think having a manager is really important, because there's lots of check boxes that need to be dealt with. Go ahead, Angela. You look like you want to say something.
I just have to tell you what a great speaker you were, how much I loved your talk. I will never think of SMAD4 again without thinking of it as whispering or yelling. I just thought your metaphors were great. Your slides were terrific. And the same applies to Mark. To the parent organizers, I can't tell you how much we're enjoying this too. This is great. Many of us have never heard each other speak about these topics, so it's wonderful.
And so emotional when you say things like that. Thank you.
This is wonderful. We're all having a great time. But in terms of how you inform your doctors, like Lisa and I are such kindred spirits. I always say to a patient, if the doctor says, I've never heard of this before, but tell me more, that's fine. If they say, I've never heard of this, but I don't really care, like Lisa said, you try to find somebody else. The next thing I always tell my patients, this is Coach Angela talking, You have to be the teachers. I say Myhre is kind of like a religion. You have to spread the word. You have to become the disciples, the apostles. You just got to get out there. Many people say, well, that's not my style. And I can appreciate that. So I really feel the Myhre Syndrome Foundation is one-stop shopping. We have a great MGH Myhre Syndrome clinic site. Feel free to visit it later on. But for one-stop shopping, you go to that Myhre Syndrome Foundation, you get pictures of people, pictures of the board, pictures of us, the international board. All the resources are there. So I think it's great. And again, you may not find a doctor who's going to be willing to sit there and doing it with you. Maybe come in and have it loaded up in your phone and just say, this is what I'd like you to know.
Right.
Yeah, I have used the handbook on several occasions. I hand the handbook to the doctors, and it's a wonderful tool to explain what it is.
Well, I get a lot of queries from around the country and around the world. Some of your doctors may have already contacted me, and I have a standard style. Number one, I cannot tell a doctor what to do. It's just the style of medicine, but I can provide them with the education. And then the education that I give them is just like a little personal greeting to just sort of outreach. And I send them the gene reviews, the Orphanet article by Dr. Valerie Cormier-Dare's group, and then the handbook. And then usually I introduce them to Kate. So we make it really a total community.
I have one last question, and we have a couple more minutes, so I will go ahead and ask it. With the increased amount of families showing an inheritance pattern, what does that mean for people with Myhre who would like to consider a family in the future? And also, should parents of children with Myhre consider getting tested?
So part of the standard of care, if we have an individual that we diagnose with Myhre syndrome, if parents are available, we do test them to confirm that. Whether it happens all the time, it's sometimes challenging, but it's recommended, especially if couples are in childbearing years. For individuals where it looks like it's de novo, and so I want to give you the de novo caveat. You know, when we test DNA, we're testing blood, and blood doesn't always reflect what's going on in our germ cells, our eggs and our sperm. And so that for an individual that's had one child with Myhre syndrome, they do have a small chance of having a second child. And they could be mosaic for that gene variant in eggs or sperm. For somebody who has Myhre syndrome, who then would like to parent a child, mostly father a child, there's options that people have. And what we do is we make referrals to our OBGYN genetics team to talk about things like pre-implantation genetic testing, where rather than conceiving naturally, somebody would conceive by in vitro fertilization. And if someone chooses, they can only implant the embryos that don't have Myhre syndrome. And I know this is hard to hear because I couldn't imagine a world without people with Myhre syndrome. My life is better because of the people with Myhre syndrome. But it is a choice that's there.
And stay tuned, everybody, for an article coming out of an international collaboration led by Bert Callaware, who's going to be speaking later, about adults with Myhre syndrome. And there are a substantial number, as people know, of families with Myhre syndrome. And it does seem like meaning a parent-to-child transmission. It does seem like these are all been currently the arginine 496 cysteine variant. Whether it could happen for somebody with another variant is unknown or present. And when parents ask me this, I usually just speak philosophically. I think, becoming a parent takes so much, right? It's not just about a variant. It's not about just biology. It's about lifestyle, independence, overall health. And I think those factors probably play a big role too.
Thank you, Dr. Schimmenti, and thank you so much, Dr. Lynn. Oh my gosh, we so appreciate both of you